Hi, my name is Ivy Meiji Romano, and in this video, we are going to discuss about the other type of immunity of the immune system, which is the adaptive immunity, or also known as the specific immunity. So for over the last 4 decades, there have been an increasing number of people who are suffering with allergies and autoimmune diseases. And the researchers and scientists have hypothesized that it is due to the inadequate development of the immune function. So now we are going to talk about the other type of immunity, which is the specific immunity or the adaptive immunity. So, what is adaptive immunity? So, it is the ability of the body to recognize and destroy certain pathogens, and the response to them improves each time the pathogen is encountered. So, it has two characteristics. So, it has already been mentioned on the definition of adaptive immunity. The first one is the specificity, which is the ability to recognize and destroy foreign invaders. And the next one is the memory, which is the ability to record or remember those pathogens, such as the microorganisms like viruses and bacteria, in order to effectively eliminate them once they enter again the human body. So that is the adaptive immunity. Next is the antigens. So what is antigens? So it is defined as any substance that causes the body to make an immune response against that substance. So the antigens are the ones who stimulate the immune response. So without the presence of the antigens, then an immune response will not occur, because they are the ones who activate the lymphocytes, which are very necessary and very crucial in order to initiate the immune response. So antigens are divided into two groups. The first one is the foreign antigens, or defined as it is introduced from outside the body. Microorganisms such as bacteria and viruses and chemicals released by microorganisms are examples of foreign antigens. The pollen, animal hairs, foods, and drugs can cause an allergic reaction because they are foreign antigens that produce an overreaction of the immune system. So tissue and organ transplants are often used to treat certain diseases. However, transplanted tissues and organs contain foreign antigens, and the response to these antigens can cause rejection of the transplant. Next one is the self-antigens. So self-antigens are molecules the body produces to stimulate an immune response. So these are the lymphocytes, the B cells, T cells, helper T cells. So the response to self-antigens can be beneficial. So for example, the recognition of tumor antigens can result in destruction of the tumor. But the response to self-antigens can also be harmful. So examples of these are the autoimmune diseases that results when self-antigens stimulate unwanted destruction of normal tissue. An example is rheumatoid arthritis, which destroys the tissue within the joints. So that is the types or the groups of antigens. Adaptive immunity can be divided into antibody-mediated immunity, which involves a group of lymphocytes called B cells and proteins called antibodies, which are found in the plasma. And the second one is the cell-mediated immunity, which involves the actions of a second type of lymphocytes called the T cells. So several subpopulations of T cells exist. For example, is the cytotoxic T cells that produce the effects of cell-mediated immunity, and the helper T cells that promote or inhibit the activities of both antibody-mediated immunity and cell-mediated immunity. This is the comparison of the innate and the adaptive immunity. There you can see the primary cells, the origin of the cells, the site of maturation, the location of mature cells, primary secretory product, the primary actions, and the allergic reactions. So the primary cells of the innate immunity are the neutrophils, eosinophils, basophils, mast cells, monocytes, and macrophages. In adaptive immunity are the lymphocytes, which are the B cells in antibody-mediated immunity, and the T cells, which is used for the cell-mediated immunity. So all of these cells are from the red bone marrow in the innate and the adaptive immunity. So the site of maturation in the primary cells are also in the red bone marrow, also in the adaptive immunity in the antibody-mediated immunity. However, in the cell-mediated immunity of the adaptive immunity, the site of maturation is in the thymus. And in the innate immunity, the location of the mature cells are in the blood, connective tissues, and the lymphatic tissues. Also, in the adaptive immunity, the location of mature cells are in the blood and lymphatic tissue, as well as in the cell-mediated immunity, the blood and lymphatic tissues. And the primary secretory products of the innate immunity are histamine, complement, prostaglandins, leukotrienes, kinins, and interferons. And in the adaptive immunity are the antibodies and the cytokines. And the primary actions of the innate immunity are the inflammatory response and the phagocytosis. And in the adaptive immunity is the protection against extracellular antigens such as the bacteria, toxins, and viruses outside of the cells, and the protection against intracellular antigens, viruses, and intracellular bacteria and tumors responsible for graft rejection. And in allergic reactions, so in the innate immunity, there is no allergic reactions. However, in adaptive immunity, there's immediate hypersensitivity and delayed hypersensitivity. So that is the comparison of innate and adaptive immunity of the immune system. Now, let's discuss the origin and development of the lymphocytes. So, the stem cells in red bone marrow are capable of giving rise to all the blood cells. So, some stem cells give rise to pre-T cells, which migrate through the blood to the thymus, where they divide and are processed into T cells. Other stem cells produce pre-B cells, which are processed in the red bone marrow into B cells. So after processing, the B cells enter the blood from the red bone marrow, and T cells enter the blood from the thymus. So as you can see in the figure, both types of cells move through the blood to lymphatic tissues. So these lymphocytes live for a few months to many years and continually circulate between the blood and the lymphatic tissues. So as you can see in the figure, both of the B cells and the T cells originate from the stem cells in the red bone marrow. B cells are processed from pre-B cells in the red bone marrow, whereas the T cells are processed from pre-T cells from in the thymus, and both B cells and T cells circulate to other lymphatic tissues such as the lymph nodes. So, small groups of identical B cells or T cells called clones form during embryonic development. So, each clone is derived from a single unique B cell or T cell. So, each clone can respond only to a particular antigen. However, there is such a large variety of clones that the immune system can react to most antigens. Among the antigens to which the clones can respond are self-antigens. Because this response could destroy the body's own cells, clones acting against self-antigens are normally eliminated or suppressed. Most of this process occurs during prenatal development, but it also continues after birth and throughout a person's lifetime. Now, let's proceed to the activation and multiplication of lymphocytes. So, the specialized B cells and T cells clones can respond to the antigens and produce adaptive immune response. So, for the adaptive immune response to be effective, two events must occur. The first one is the antigen recognition by the lymphocytes, and the second one is the lymphocyte proliferation. So now let's discuss first the antigen recognition. So lymphocytes have cell membrane proteins called the antigen receptors on their surfaces. So the antigen receptor on B cells are called B cell receptors, and those on T cells are called T cell receptors. So each receptor binds with only a specific antigen. Each clone consists of lymphocytes that have identical antigen receptors on their surfaces. So activation of the lymphocytes of a particular clone occurs when antigens combine with the antigen receptors of a clone. So once lymphocytes are activated, the adaptive immune response begins. So in many cases, the antigens that activate the lymphocytes are actually a portion of larger molecules. So B cells and T cells typically recognize antigens after these large molecules have been processed or broken down into smaller fragments. So antigen presenting cells such as the macrophages process the larger molecules and present antigens to B cells and the T cells. So the larger molecules are taken into macrophages by phagocytosis, which are mentioned earlier by my group mate, and are broken down into smaller antigen fragments, and the processed antigen fragments are bound to major histocompatibility complex molecules transported to the cell membrane of the macrophages and presented to B cells and the T cells. So major histocompatibility complex or MHC molecules are the glycoproteins that have binding sites for the antigens. So different MHC molecules have different binding sites, that is, they are specific for certain antigens. So it is very important not to confuse the MHC molecules with the antigen receptors being described earlier. Though both types of receptors interact with antigens, MHC molecules are a different group of receptors found on the membrane of many types of the cells. So there are two classes of the major histocompatibility complex. The first one is the MHC class 1 molecules and the MHC class 2 molecules. So the first type, which is the MHC class 1 molecules, are found on the membranes of most nucleated cells, and the MH2 molecules are found on the membranes of the antigen presenting cells, B lymphocytes, and other defense cells. So that is the major histocompatibility complex or the MHC molecules. Now let's discuss the second event for an effective adaptive immune response to occur, which is the lymphocyte proliferation. So it is a very important process that generates the needed defense cells to protect the body. So it increases the number of T cells and B cells, but the processes of these lymphocytes differ for each lymphocyte class. So first is the proliferation of the helper T cells. So the helper T cell responds by producing interleukin-2 and interleukin-2 receptors. So interleukin-2 binds to the receptor and stimulates the helper T cell to divide. So the daughter helper T cells produced by this division can again be presented with the antigen macrophages and again be stimulated to divide. So thus, the number of helper T cells is greatly increased. So it is very important for the number of helper T cells to increase because these helper T cells are necessary for the activation of most B cells or T cells. So for example, B cells have receptors that can recognize antigens. So most B cells, however, do not respond to antigens without stimulation from helper T cells. So without functional helper T cells, the immune response of B cells would not be effective to prevent disease. So now let's proceed to the B cell proliferation. So it begins when a B cell takes in the same kind of antigen that stimulated the helper T cell. So the antigen is processed by the B cell and presented on the B cell surface by an MHC class II molecule. So a helper T cell is stimulated when it binds to the MHT class II or the antigen complex. So there is also co-stimulation involving CD4 and interleukins. So as a result, the B cell divides into two daughter cells. So these daughter cells may differentiate into plasma cells, which produces antibodies. So the division process continues, so eventually producing many cells capable of producing antibodies and resulting in sufficient antibodies to destroy all the antigen. Now let's discuss the anti-mediated immunity. So exposure of the body to an antigen can lead to the activation of B cells and the production of antibodies. So the antibodies bind to the antigens, which can be destroyed through several different mechanisms. So because antibodies are in body fluids, anti-mediated immunity is effective against extracellular antigens such as bacteria, viruses, and toxins. So it is also involved in certain allergic reactions. Next is the structure of these antibodies. So antibodies are proteins produced in response to an antigen. So they are Y-shaped molecules consisting of 4 polypeptide chains, 2 identical heavy chains, and 2 identical light chains. So the end of each arm of the antibody is the variable region. So this region is the part of the antibody that combines with the antigen, and the variable region of a particular antibody can join only with a particular antigen. So this is similar to the lock and key model of the enzymes. So the rest of the antibody is the constant region, and it has several functions. So for example, the constant region can activate complement, or it can attach the antibody to cells such as macrophages, basophils, and mast cells. So antibodies can make up a large portion of the proteins in plasma. So this is the structure of the antibody. So as you have noticed, it is the Y shape. So there you can see the uh, antigen binding site, the heavy chain, the light chain, the constant regions, and the variable regions. So next is the effects of antibodies. So the effects of antibodies can directly or indirectly affect the antigens. So direct effects can occur when a single antigen binds to an antigen and inactivates the antigen, or when many antigens are bound together and are inactivated by many antibodies. So the ability of antibodies to join antigens together is the basis for many clinical tests such as blood typing, because when enough antigens are bound together, they form visible clumps. So next is the indirect effect of the antibodies to the antigen. So after an antibody has attached by its variable region to an antigen, the constant region of the antibody can activate other mechanisms that destroy the antigen. So for example, the constant region of antibodies can activate complement, which stimulates inflammation, attracts white blood cells through chemotaxis, and lyses bacteria. So when an antigen combines with the antibody, the constant region triggers the release of inflammatory chemicals from mast cells and the basophils. So people who have hay fever inhale the antigens, which are then absorbed through the respiratory mucous membranes. So the combination of the antigen with antibodies stimulates mast cells to release inflammatory chemicals such as the histamine. Next is the antibody production. So the production of antibodies after the first exposure to an antigen is different to the second exposure or the subsequent exposure of the antigen. So the primary response is the result from the first exposure of a B cell to an antigen. So the B cell proliferates to form plasma cells, and these plasma cells produce then antibodies. Next is the secondary response of the antibody production, which occurs when the immune system is exposed to an antigen against which it has already produced a primary response. So the memory B cells are responsible for the secondary response. So when exposed to the antigen, the B cells quickly divide to form plasma cells, so which rapidly produce antibodies. So the secondary response produce better protection than the primary response for two reasons. One is the time required to start producing antibodies is less, hours to a few days, and two, more plasma cells and antibodies are produced. So as a consequence, the antigen is quickly destroyed, no disease symptoms develop, and the person is immune. So we are done with the antibody-mediated immunity. Now let's proceed to the cell-mediated immunity. So cell-mediated immunity is the function of cytotoxic T cells and is most effective against microorganisms that live inside body cells. So it is also involved with some allergic reactions, control of tumors, and graft rejection. So cell-mediated immunity is essential for viral infections. So when viruses infect the cells, they direct the cells to make new viruses, which are then released to infect other cells. Thus, cells are turned into virus manufacturing plants. While inside the cell, the viruses have a safe haven from antibody-mediated immunity because antibodies cannot cross the cell membrane. And then the cell-mediated immunity fights viral infections by destroying virally infected cells. So when viruses infect the cells, some viral proteins are broken down and become processed antigens that are combined with MHC class I molecules and displayed on the surface of the infected cells. So cytotoxic T cells can distinguish between virally infected cells and non-infected cells because the T cell receptor can bind to the MHC class I or viral antigen complex, which is not present in uninfected cells. So the T cell receptor binding with the MHC class I or antigen complex is a signal for activating cytotoxic T cells. So co-stimulation by other surface molecules such as the CD8 also occurs. So the helper T cells then provide co-stimulation by releasing cytokines such as the interleukin-2, which stimulate activation and cell division of cytotoxic T cells. So increasing the number of daughter helper T cells results in greater stimulation of the cytotoxic T cells. So in cell-mediated responses, helper T cells are activated and stimulated to divide in the same fashion as in the antibody-mediated immunity. So, after cytotoxic T cells are activated by an antigen on the surface of a target cell, they undergo a series of divisions to produce additional cytotoxic T cells and memory T cells. So, the cytotoxic T cells are responsible for immediate cell-mediated immune response, and the memory T cells provide a secondary response and long-lasting immunity in the same fashion as the memory B cells. So the cytotoxic T cells have two main effects. One is they release cytokines that activate additional components of the immune system. So for example, some cytokines attract innate immune cells, especially macrophages. So these cells are then responsible for phagocytosis, of the antigen and the production of an inflammatory response. So cytokines also activate additional cytotoxic T cells, which increases the effectiveness of the cell-mediated response. And the second one is that the cytotoxic T cells can come in contact with other cells and kill them. So virally infected cells have viral antigens, tumor cells have tumor antigens, and tissue transplants have foreign antigens that can stimulate cytotoxic T cell activity. Thus, the cytotoxic T cells bind to the antigens on the surfaces of these cells and cause the cells to lyse. Now let's discuss the third type of immunity, which is the acquired immunity. So active immunity results when an individual is exposed to an antigen and the response of the individual's own immune system is the cause of the immunity. So passive immunity occurs when another person or an animal develops immunity and the immunity is transferred to a non-immune individual. So active immunity has two types. First is the active natural immunity. So this results from natural exposure to an antigen such as the disease-causing microorganisms that stimulates the immune system to respond against the antigen. Next is the active artificial immunity. So it is when the antigen is deliberately introduced into an individual to stimulate the immune system. So this process is called vaccination, and the introduced antigen is the vaccine. So a vaccine is usually administered by injection. So examples of vaccinations are the DTP injection against diphtheria, tetanus, and pertussis, and the MMR injection against mumps, measles, and rubella. So the vaccine usually consists of part of a pathogen, either a dead microorganism or alive or the altered one. So the antigen has been changed so that it will stimulate an immune response but will not cause the disease symptoms, because active artificial immunity produces long-lasting immunity without disease symptoms, so it is preferred method of acquiring adaptive immunity. Now let's proceed to passive immunity. First one is the passive natural immunity that results when the antibodies are transferred from a mother to her child across the placenta before birth. So during her life, the mother has been exposed to many antigens, either naturally or artificially, and she has antibodies against many of these antigens, which protect her and the developing fetus against disease. So some of the antibodies can cross the placenta and enter the fetal blood. So following birth, the antibodies protect the baby for the first few months. Eventually, the antibodies break down and the baby must rely on its own immune system. If the mother breastfeeds her baby, antibodies in the mother's milk also provide some protection for the baby. So next is the passive artificial immunity. So it involves the collecting of antibodies from one source and introducing them to an infected individual, usually through injection. So achieving passive artificial immunity begins with vaccinating an animal, such as a horse. So after the animal's immune system responds to the antigen, antibodies are removed from the animal and injected into the human requiring immunity. So alternately, a human who has developed immunity through natural exposure or vaccination can serve as a source of antibodies. Passive artificial immunity provides immediate protection because the antibodies either directly or indirectly destroy the antigen. So passive artificial immunity is therefore the preferred treatment when not enough time is available for the individual to develop his or her own active immunity. However, the technique provides only temporary immunity because the antibodies are used or eliminated by the recipient. So that is the acquired immunity, which is also necessary for an effective immune response. So that ends our report about the immune system. Thank you so much for listening, and God bless us all.